Transcription is RNA synthesis. So we're going to convert from DNA base to RNA base, but like replication, transcription is only and always 5' to 3'. That means exactly the same thing. It means adding on new nucleotides to the 3' end of a bond transcript. And just like replication, it's anti-parallel. So 5' to 3' transcription means that the template is 3' to 5'. Let's take a look at the nucleotides, the building blocks that we're dealing with. We have a whole new set of building blocks, but they will look very familiar. So first of all, instead of the deoxyribose, we now have the full ribose sugar. As we start transcription, the first nucleotide is always A. That's the first nucleotide put in ATP, and so invariably in almost all cases, so transcripts that have just been made start with a triphosphate. And from the ATP, the process is exactly the same as replication. Two key differences between what we've just done with DNA and what we're now doing with RNA is that first, the sugar is the full ribose sugar. And secondly, we substitute uracil for thymidine. So uracil is just like thymidine, except we don't have the methyl group there. These are the only two differences between DNA synthesis that we've been doing and RNA synthesis. We make the phosphodiester bond exactly the same way. That is, we remove water and we make this phosphodiester bond, it's kind of crooked, but it's the exact same phosphodiester bond made exactly the same way by removing water and taking off this pyrophosphate. So the synthesis process, the mechanism of converting the monomer building blocks, the ribose nucleotides, into a polymer is exactly the same. The enzyme that does this is RNA polymerase. 5' to 3', adding on nucleotides. So here's your 5' end. Here's your 3' end. We will continue to add nucleotides to that 3' end exactly the way we did it for DNA. Over here is a DNA template. So the process is exactly the same. two key differences, one in the sugar, and we change thymidine for uracil. Any questions on how we make RNA? Yes?
So making a strand, they remove one set of the phosphate?
Louder.
Is it, so it goes from being a triphosphate to a monophosphate?
Nope. Nope, I have left this alone.
I'll talk about the circled part.
Oh, yeah, it's the phosphodiester bond. COPOC. Just exactly what we did for DNA. That does not change. It is the exact same phosphodiester bond. You look puzzled.
Well, so what is the part where you circle? It's removing that?
So
have the circle
we have to
here.
get rid of that.
Okay.
Someone else have a hand? Okay. Anybody else? Yes.
What does the RNA polymerase do again?
RNA polymerase makes RNA. It takes the nucleotides and polymerizes them into a polymer, the building blocks into a polymer.
Are there multiple RNA polymerases
We're
or just
going to talk about that. There are, in some organisms, not others. And so remember, you've already used primase, which is an RNA polymerase, but the primase works in replication. This is transcription. This is another whole process. Enzymes are different, but primase does exactly the same thing. Just makes short primers. Anybody else? All right, so let's take a look at DNA. So we're going to use double-stranded DNA. We're going to do this both in bacteria and in eukaryotes. We can make a copy here, 5' to 3'. That means the template is 3' to 5'. Template is anti-parallel. So we can make an RNA copy there. We can make RNA copy there. And that's all we can do. So we cannot do that. There is no enzyme that can copy the opposite direction. Again, the DNA polymerase and the RNA polymerase work exactly the same. That is, 5' to 3' is the only direction that they can synthesize. So as we take a chromosome, and you've looked at lots of chromosomes and genes on chromosomes, you've seen that a chromosome map of genes has some genes with the arrow pointing right and some genes with the arrow pointing left. So now what you see, the reason for that is that the polymerase can only transcribe in one direction. So this is, in other sense, a gene like that. The other one is a gene like that. This shows more detail than that, but this is exactly the same thing. We are constrained to transcribe to the right on the top strand, transcribe to the left on the bottom strand. And we can set up all sorts of combinations. So here we have two units converging on each other. Here we have two units diverging on each other. So as an RNA polymerase is looking for genes, it needs the same thing that the ribosome needs. It needs to be able to find start codons. So you know the start codon is AUG, and that tells the ribosome where to start. You have a set of 3 stop codons that tells the ribosome where to stop. We have a similar set of instructions. So those are instructions embedded in the RNA. Now we have instructions embedded in the DNA. And now the start is called a promoter. And we abbreviate that with P. The stop is called a terminator. We abbreviate that with little t. And so we have promoter, terminator, promoter, and terminator. And so these are the signals, sequences in the DNA that the RNA polymerase is looking for to tell it not just where to start. So here we have 2 promoters. So the polymerase knows that there is a start of transcription, but this promoter says use the top strand and go to the right. This promoter says use the bottom strand and go to the left. And so know that the 2 strands do not have the same sequence. They're complementary. They are not identical. So we've already looked at translation. We've looked at the codons, start and stop. Translation is AUG. Stop is UAA, UAG, UGA. Now we have translation. Start and stop is promoter and terminator. So we have a different set, a completely different set. This is transcription. We are going to look at promoter sequence this morning, or this afternoon. We're going to look at promoter sequence. We are not going to look at the terminator sequence. But these are really short sequences. So start and stop, we have 2 different sets of start and stop signals. One set is in the DNA for RNA polymerase. The other is in the RNA or the ribosome. And you've got your codon dictionary. In fact, you've been using your codon dictionary. That's what these are all about. And now we're going to switch over and look at these. Any questions on this introduction to transcription?
You're saying or you're saying like that as an RNA is what? So you need to find a start codon?
RNA polymerase?
An RNA polymerase. And what it does in order that it would require finding a start codon?
Ribosome?
An RNA is finding what or doing what exactly?
I have no idea what you're asking.
You're asking what comes before you need to find a start codon and after the as an RNA polymerase, that's what?
I have no idea what you're asking. What do you want to know?
I just want to know like what goes in between that sentence.
I have no idea. I'm not listening to myself. You want to ask a question about what's on the floor? Okay. Anybody else? Any questions? We have 2 different sets of start and stop signals. Let's see how this works. So now we're going to take and do another set of maps. We have chromosome maps and gene maps. The chromosome maps are huge. Gene maps are much smaller. Now as we start to look in at the details in the gene map, we have a different set of symbols. So the symbols are that this bent arrow tells you where the start of transcription is. We have a different set of numbers. So you've seen that gene maps have different coordinates than chromosome maps. And now we're looking in at a gene map. Everything to the right inside is given plus numbers. So we have plus 20, plus 40, and so on. Everything to the left of the arrow is given minus numbers. And so we have minus 10, minus 20, minus 30, minus 40. The promoter sequence is just outside the start of the transcript.
And this is just an RNA, if this model was used, or this also used?
Well, so that is the DNA.
Okay.
And so this is a set of coordinates. Now we're magnifying that region that we had a gene map before, like one of the ones above. We're magnifying that region so we can zoom in on sequence details to see what exactly the promoter sequence is.
So until the start, like the promoters, they're not negative and positive. It's only when that bar is there.
Right, so in going from a chromosome map or to a gene map, gene map's going to be the same as this, but we're just zooming in. And so this is a new symbol. We haven't used this symbol before. Anybody else? All right, so we're going to look at the promoter. The promoter has two small 6-base sequences. One at this location, TA box. It's a 6-nucleotide sequence called a Tata box. Very easy to pronounce the actual DNA sequence because the actual DNA sequence is a whole set of variants based on TA. And so you can vary TAA, TAA, or TATAAA, a whole set of sequences, but it's called a Tata box. And that Tata box is right around minus 10. At minus 35, we have a minus 35 box, because you can't pronounce a 6-base sequence that's mostly G's and C's. This is the promoter. This is it. Two small 6-base pair sequences define the start of transcription. The Tata box and the minus 35 box. Now we have this concept called a consensus sequence. And a consensus sequence means that somebody sat down with 100 different promoters and they listed all those Tata box sequences and came up with what is the most common first base. The most common first base, about 95% of the time, is T. What is the most common second base? A, and so on. It doesn't mean that every promoter has to have exactly the same sequence. In fact, there are good promoters and poor promoters because the promoter sequence can vary considerably and still be a variation on TATA, 6-base variation on TATA. So a good promoter is a sequence that has a very high level of getting the RNA polymerase onto the gene and making RNA. A poor promoter is a sequence that isn't so good. The RNA polymerase has to struggle. You can't get as much RNA synthesis from a gene with a poor promoter as you can from a gene with a good promoter. And that's a really important concept because cells do not need to have all their genes transcribed at the same rate. Some genes you might want a lot of RNA from, other genes you just want a small amount, and so you can control that through the promoter sequence. Everybody understand what I mean, this concept of poor promoters, good promoters?
Not about that, but for the negative 35 box and the TATA box, so it'll find the negative 35 box and be like, there's going to be a TATA box soon.
No.
So will it find the negative 35 box and then it knows that there's going to be a TATA box very soon?
And actually, that's exactly the way it works. What I'm going to show you is the RNA polymerase looks for the minus 35 box first, and then it just sort of falls down on top of the TATA box. But that's identifying which strand and which direction.
Is the arrow where RNA synthesis begins?
The arrow is where it begins, and so it begins at base 1. There is no 0 in the coordinates. So it's minus 1 and plus 1. Yes?
You said that a good promoter is one that simply has a high instance of RNA binding and transcription.
So functionally, yes, a good promoter is one that has a high rate, makes a lot of RNA from that promoter, as opposed to a poor promoter. And a good promoter is also one that most closely matches the consensus. But oddly enough, there's no promoter, as far as I know, that is a perfect match for the consensus. So there's a lot of variability there. Yes? So consensus sequence means that people have taken hundreds of different promoters, lined them all up, and asked, what is the most common first base? It is T. About 95% of all promoters will start with T. What is the most common second base? Most common second base is A, a little bit less than 90% perhaps. Third base, what's the most common base? It is T. And from then on, you have a mixture of A's and T's. It's not as highly constrained. So consensus just means people take a vote. What's the most common base here for 1, 2, 3, 4, 5, and 6?
So first is T, second is A, third is T.
Yep. Anybody else? All right. So from this whole region, though, is called the promoter. The sequence in between the minus 35 box and the TATA box, the sequence doesn't matter. So if you start making mutations and go down the line and make mutations, what you find is that if you change the minus 35 box or change the TATA box, the promoter activity drops drastically. But you can change any bases in between with zero effect, as long as you don't add or subtract the base. And so the DNA in between the minus 35 box and the TATA box, the sequence doesn't matter, but the spacing is important. The spacing has to match the dimensions of the RNA polymerase. The same is true out here for the terminator, but I'm not going to give you that sequence. So we have this unit from promoter to terminator called a TU, or transcription unit. So this is a unit of transcription. It has a start promoter and a stop terminator. Now, most of your transcription units code for protein, but not all, only most. And so for the average transcription unit, what we have nested inside is an ORF, an open reading frame. We've already covered this in the code. The open reading frame starts with a start signal and ends with a stop signal. So now we have some more details on what a protein coding gene looks like. A protein coding gene has an open reading frame nested inside a transcription unit. So this is the physical features, and to the left here it's obviously expanded, not in the same scale. The physical features of a transcription unit. Any questions so far? All right. So this is bacteria. Bacteria has one single RNA polymerase. And it may sound funny to say that because you know that primase is an RNA polymerase, but that's still what people say, because primase does not participate in transcription. Remember, primase is a dedicated component of replication. So one E. coli RNA polymerase handles all of the transcription of all the bacterial genes. That's different from the way you handle the situation. So we'll see that coming up. One RNA polymerase, the way the RNA polymerase works, RNA polymerase looks for the -35 box. That means that the E. coli RNA polymerase can read DNA. So it has a protein subunit that can read the sequence of DNA. And the -35 box and title box are set up so that there's directionality. The polymerase sits down the -35 box and then just sort of folds down on the gene. So here is the RNA polymerase. Notice the RNA polymerase overlaps the start of transcription. It's not going to move very much. It's going to stay there while it starts the process, but it has to melt out the promoter and make a bubble, kind of like replication, but kind of not. Because we're not going to make a pair of Y forks that diverge. We're going to make a small bubble, and then the polymerase is going to move that bubble along the gene. And so we're going to make the bubble starting right at the title box, and that bubble is going to encompass the start of transcription, and then we're going to put an ATP in, because in virtually all cases, transcription starts with A as the first nucleotide, and then whatever the template specifies for the second, third, and fourth. So a relatively simple, straightforward mechanism for how bacteria start transcription. There's one polymerase that does all transcription units in E. coli. Any questions on this, on the terminology?
Yeah, so then what happens with the title box there? After,
What do you mean what happens?
with the asterisk center?
Oh, so that, I can just put that there and say this is where melting starts. So melting starts here, and it expands into the start of transcription. And the polymerase doesn't move for a little bit. It's going to sit there as it makes a very short RNA right where it is. And then it starts to move. And the bubble moves, not like replication. Anybody else? Yes?
So the RNA starts at the top box, you said? And
The bubble starts at the top.
doesn't go to the bubble, RNA bubble.
Well, that's where it melts out the DNA. Why do you think it melts out the DNA in the powder box?
So it doesn't.
Is it so I could read the base pairs?
No.
Is it so I could read the base pairs?
Well, it reads the base pair, but why the powder box?
So it has enough room to like quickly move it up, and before it has to start like adding.
Yeah, but why the powder box?
It's easier because they have two hydrogen bonds.
Yeah, louder.
It's easier because they have two hydrogen bonds.
Powder. It's pure T's and A's. They're all 2 hydrogen bonds. G's and C's are 3 hydrogen bonds. And so the Tata sequence is the easiest to melt. Remember, in replication, there was a DNA A that twisted open the ori and a helicase that moved it. Here, RNA polymerase is doing everything by itself. RNA polymerase itself melts out and opens up the DNA. DNA polymerase can't do that. And so the reason that we have TATA, 6 bases of that, is because it's easier to pull apart than G's and C's. Anybody else before we switch over to look at your genes? Don't leave that. You can leave that up. So now as we switch over, first of all, again, E. coli has 1 RNA polymerase that transcribes all of its genes. You have 3 RNA polymerases. Pol 1 is for ribosomal RNA. Pol 2 makes your message. And pol 3 makes small RNAs like transfer RNA. So you have a division of labor that bacteria does not have. But everything that we talked about in terms of the mechanism of transcription is exactly the same, 5' to 3', except for one thing. This is a DNA template. Your template is chromatin. And so that makes a huge difference in how transcription occurs. Let me set some chromatin up for you. And so before we can even think about transcribing a gene, we may even have to take it from this 100 angstrom fiber and convert it to the beads on a string form. And then we have to remodel, that's the term, remodel the chromatin so that we have the promoter now in open essential step. They have to do all kinds of things because of chromatin. Every time you do anything on DNA, whether it's replicate, whether it's repair the DNA, whether it's recombination, whether it's transcription, you have to take into account the fact that you've got nucleosomes and you've got to move the nucleosome. So in order to remodel, what you have to do is modify the histones. This, going from the 100 angstrom to the beads on a string, is all about H1. Remember that H1 is not in the octamer. And so, way more complex to do transcription, to do anything on your DNA than in bacteria. By modifying the histones, we add phosphate, we add acetate, we add methyl groups, all sorts of things, modifying the histone proteins so that the chromatin and nucleosomes can be moved aside until we finally have that opened up.
Did you say that we remove H1?
So H1 is the key to going from the fibers, the 100 angstrom, 300 angstrom fiber, to the beads on a string. And so H1 is really mediating nucleosome-nucleosome interaction. And these nucleosomes are interacting with each other due to H1.
So H1 is phosphorylated, so it,
Well, I don't want to go into the details. The H1 is controlling it. Let's just leave it at that. Anybody else? So the basics of the promoter is going to be pretty similar, but everything that you do compared to bacteria, you do with bigger things, not necessarily better. So we still have the start of transcription, and we still have a Tata box, except that the Tata box is now out at about -30. And interestingly, I said that the location is absolutely fixed in bacteria. It really doesn't matter very much where your Tata box is. So in some genes, it's a little bit closer, other genes, it's a little bit further. In bacteria, the Tata box and the 35 box, that's the only sequence that's important. There's a T at the start of transcription, because A is the first base. But here, the Tata box can be moved, so we have to identify the start of transcription, a small region called INR, which just stands for initiation region. And so because the Tata box isn't fixed location, we have to have another element to say, okay, here's where I want you to start. And then we have a third element inside called DPE, which is downstream promoter element. So bacteria have two elements. They are rigidly fixed in place. The distance between them is rigidly fixed. That's not the case with your genes. You have 3 elements, and the way it works is a promoter can be any 2 out of 3. What that means is that a lot of your genes don't even have that canonical Tata box. So probably 2/3 of your genes has the Tata box, but a third doesn't. A third just uses the INR and DPE to specify where to start. So we see a whole bunch of differences between the top board and this one. We see a larger promoter spread out over a much larger distance And now we see 3 elements instead of 2 elements.
Did that say LNR or INR?
INR,
INR.
initiation region. That's what it stands for. Anybody else? Yeah.
And you were saying that the Tata box can be free floating, or not free floating, but it doesn't necessarily have to be in one specific.
Right, so if you look at 100 genes, you'll find it in different positions within about 20, 30 base pairs.
When you say 2/3, meaning the third one like is missing, or it just doesn't use it? Or-
No, it's missing. So what's called housekeeping genes, like RNA polymerase, DNA, those are genes used in every single cell. Housekeeping genes very often do not have a Tata box at all. It's just a common trait.
Do eukaryotes have a negative 35 box or no?
They do not have the -35 box. No -35 box. There can be other DNA sequences, but they're not part of what's called the core promoter. I don't want to get into that too much.
So would 2 out of 3 be a variation of any of those 3?
Any 3, yeah, you have any combination, 2 out of 3.
Tata and a DPE?
Tata DPE, INR DPE, Tata INR, yeah.
With the sequence between, are they always going to be in between like nucleosomes or?
No. Oh, you mean these? Yes. So invariably, what happens when you move things away, you're just clearing out the promoter. And so that typically puts nucleosomes flanking the promoter until we assemble the RNA polymerase, and then we'll probably get rid of that. But we don't have to. We can continue to move it aside. We'll talk about that. I don't know if we can get to it today.
What does LNR stand for?
INR.
Oh, INR.
Let me change that. Make it much smaller. There. Initiation region. Initiation region. Anybody else? Okay, so we see a bunch of differences. You've got a bigger promoter. You have 3 elements. The next huge major difference is your RNA polymerases. All 3 of them are illiterate. Your RNA polymerases cannot read DNA sequence. I said the RNA polymerase in bacteria reads the sequence, finds the -35 box, and sits down on it. Proteins, some proteins can read DNA and RNA sequence. Those are gene regulators. Yours is illiterate. Could not find a promoter if it was bumping into it. Cannot read. So we have to have a totally different mechanism to get your RNA polymerase to the right spot so it ends up looking exactly like that. And the way we do that is putting on a bunch of proteins called transcription factors. And the terminology gets messy here, but I didn't make it. We've already used transcription factor in the signal transduction pathway. The MYC gene that was activated and went into the nucleus to turn on genes is called a transcription factor. But it's a different type of transcription factor. Those are regulatory. The terminology just got put in place before people realized what was happening. So these are also called transcription factors, but they are transcription factors for the promoter. They do not regulate the gene, they just set up the RNA polymerase. So as we go through, when we talk about regulation, we'll talk about transcription factors again. Just keep in mind there are 2 very different sets. One that regulates and one that starts, and one set that starts transcription. I apologize for that. It's just an accident of history. So what we do is build platform. You're going to build a platform of transcription factors. And now, your RNA, and so by the way, so we're talking identical for 1, 2, and 3. All the polymerases are exactly identical in the mechanism by which the RNA polymerase locates a gene and starts transcription. Yes? So the one set like MIG stimulate and turn on transcription. Another set here builds a platform because your DNA, your RNA polymerase can't find the promoter, but it recognizes this DNA transcription factor platform. And now we can do the same thing. Let me do the same. And so now RNA polymerase binds to this platform, and now the exact same thing happens. It's going to open up the DNA. If there's a title box there, usually at the title box. If not, there'll be some other easy sequence to open up. And start off pretty much the same way as bacteria, but just a totally different mechanism for a lot of reasons than bacterial transcription. Yes?
So we're going to do along with the again with the bubble.
Yes, so the bubble is going to be a fixed size, and the RNA polymerase moves the bubble. Replication happens much faster because the DNA polymerase only makes phosphodiester bonds. RNA polymerase has to move itself along the template. Does it know what? Oh, no. We're talking two different kinds of read. So it can make a base pair. What I'm saying is Box is closed DNA. It is a base pair. It can't read that. And so transcription factors of all flavors can read DNA sequence in base pairs. Polymerase can make base pairs, but you can't read the sequence hot in the DNA. That's what I mean by that. Everybody clear on that? Any last questions on starting this off? I'm sorry, yes.
So on the second one for eukaryotes,
Yes.
the RNP bubble covers the arrow too.
Well, this isn't a bubble. So this is the RNA polymerase. That's
Yeah,
all.
but it covers the arrow.
Oh, absolutely. It's got to cover the arrow because ultimately that's where we're going to start transcription. Yes.
So now let's say you're missing the Tata box and you have the INR and DP. Is that RNP just not going to cover that Tata area?
No.
Okay.
Actually, the platform is going to be exactly the same. The difference is whether it starts the Tata box or starts with the DPE.